Hello, I'm Dr. Georgia Ward-Fear. I'm a herpetologist, a conservationist, and I'm also a postdoc in Rick Shine's lab at Macquarie University. I'd like to acknowledge the Indigenous traditional owners whose land we're on today and pay my respects to their elders, past, present, and future. It's so great to be here with you all today to be able to share my research on the conservation of apex reptilian predators in Northern Australia. When I was a little girl, I was obsessed with dinosaurs, and I'm sure many of you can relate to that. I was particularly interested in the Megafauna of Australia. We had huge crocodiles and goannas and large carnivorous marsupials like the Thylaca leo, and they were the apex predators at the time that regulated the system from the top down, but they went extinct many thousands of years ago. These are their ancestors, the apex predators of Australia today. We still have crocs and snakes and goannas and carnivorous marsupials, but they're a lot smaller and many of them are under threat. In some ways, we're facing the second wave of loss of apex predators. They're under threat from things like land clearing, habitat degradation, disease, but also invasive species. Invasive species are the leading cause of extinction in Australia, and we have so many. We've got predators, animals that degrade the habitat with their hard hooves, animals that spread disease, but one invasive species has really captured the minds of the Australian public.
From a strange, savage land comes a hero unlike any you've ever known.
1935, 102 cane toads are shipped to Australia to eliminate the beetles that are destroying the sugarcane crops. By the look of things, this sturdy fellow is ready to go to work. So 3 cheers and welcome, brave champion.
Well, I doubt the IQ of those people. I really do, in all due respects. Toad did nothing whatsoever to eliminate the beetle problem.
The female can lay 30 to 40,000 eggs twice a year.
A toad's a nomad. That's what he does. He just keeps expanding.
You couldn't count the numbers. There's thousands and thousands and thousands.
Maybe he's eating himself out of house and home and has to keep moving.
Well, what is going to stop it? I mean, what are they going to be like? They'll be as big as camels.
And I found that they were highly toxic.
He's nothing but vermin. I think there should be a bounty put on his head.
Toads are public enemy #1. People should try and kill as many of them as they possibly can by any means that they can.
What we do is we put them straight into a bag in the freezer.
I thought about things like flamethrowers and aerial bombing.
Certainly, if the toads come to the territory, you should expect some tough treatment.
I've got a bloody toad in my freezer.
Oh, the invasion's going to go on. They're not going to stop it.
I think of the Second World War where you hear about those 8 of them out there against 500,000 on the other side.
They're actually wasting time.
Depart from here, you cursed creatures, into everlasting flames, prepared for the devil and all his bloody angels.
They're not going to stop the cane toad.
Yes, it's the cane toad, Rhinella marina. Never has an invasive species captured the Australian psyche so much. And that is a cult classic movie made a few years ago that chronicles the relationship that we've had with this invasive species. But who is the cane toad and why were they brought here? Toads actually have a history of purposeful introduction for use as a biocontrol. This is a drawing of a 19th century market in France, and it's a toad market. People would go there, buy toads and release them into their gardens to control their unwanted garden pests. Sugarcane farmers cottoned onto this and tried to adopt this strategy wide scale. Toads are originally from South America, but sugarcane farmers quickly began introducing them into Jamaica, Cuba, Hawaii, and in 1935 into Australia. They were introduced to control the larvae of sugarcane pest beetles. But they're highly fecund, they breed in big numbers, and they were introduced, they didn't control the beetles, but they began spreading. But how has a forest frog from the Amazon adapted to the dry Australian continent? Well, actually quite well. In the span of one human lifetime, cane toads have rapidly evolved to adapt to the different environments that they now inhabit in Australia. They've dispersed further than we ever could have thought and into environments that we never thought that they could live in. They've dispersed quite far into the desert, and the animals there have the ability to cope with really arid conditions. The adaptations that we see on the northern invasion front are particularly spectacular. Toads on the front have evolved longer legs to help with dispersal into new habitats and access to new resources. And this takes a toll on them. They actually develop arthritis, as that picture shows there with a lump on the spine. And down south, they've adapted to live at high altitudes and in colder climates. And despite the fact that cane toads have an ecological impact, which is considered very bad, the story of the cane toad invasion in Australia and the rapid evolution has actually been quite fascinating. There is a lot of hysteria around toads and the impacts that they exert on native ecosystems, but it's a little bit more complex than that. Some animals actually benefit from the arrival of cane toads. Cane toads possess huge toxin glands on the back of their heads, which are full of potent toxins that are unique to the toad family. Some animals have learned what parts are safe to eat, such as crows, but then there are other animals, such as the colubrid snakes, that have a pre-evolved history with toads, with the toad family in Asia, and they are able to actually consume cane toads because they have a physiological resistance to their toxin. Many of our native animals have actually learnt to live with cane toads. This is because they just don't engage with cane toads at all, or because they are accessing life history stages that are not very toxic. They've had a non-lethal experience and they've learnt from that experience and avoided toads ever after. For some species, impacts vary. We see them decline in some places, but survive perfectly fine in others, and we're not really sure why. Crocodiles are a good example of that. We expected them to crash in Lake Argyll, and they didn't. Nothing really seemed to happen when the cane toads arrived. Frillneck lizards decline but are starting to recover, and there's kind of anecdotal evidence that cane toads and pythons interact, but there's not that many records of that happening, and definitely no records of population impacts. We think that the varied impacts on these species has to do with their local ecology and the resource availability throughout the year. But for a handful of species, cane toads are an ecological disaster. These are the frog-eating predators in the environment. They decline in huge numbers. Many places go locally extinct. And the issue with these animals declining is not only is it important to preserve them for their own species' sake, but because they're the apex predators that regulate the food web from the top down, declines in these animals trigger ramifications through the whole web. They destabilise the food web. So it's important to keep them around for ecosystem integrity. Out of all of those species that I just showed you, the varanid group is hit really hard. And in particular, this species, Varanus panoptes, the yellow-spotted floodplain monitor. I'm particularly fond of this species because I worked with them for my PhD and I continue to work with them. I've done a lot of research with them. They're a large-bodied generalist predator, 2 meters long. They can get up to 4, 5, 6 kilos, the males, and they're iconic in Northern Australia. They've got a really wide distribution, and you can see from the map that the projected cane toad distribution is going to nearly engulf that of Panoptes, and so that spells trouble for these guys. In many areas behind the front, they've gone locally extinct, and in areas where they still persist in very small numbers, it's taken them 80 years and they still haven't recovered. We do see some populations persisting in places like close to the coast, and we suspect it's got to do with the food subsidies that they get throughout the year that kind of maybe limit their reliance on toads or on food at that time of year when toads are really prevalent. But this species is in trouble. I'm going to take a little sidestep to indulge my love of the varanid family, but this is a herp meeting, so I could do that. The varanids are a really remarkable family of reptiles. They first evolved in the Upper Cretaceous, 100 to 66 million years ago, and they really haven't changed much since. So they're living dinosaurs, if you like. The body plan has stayed the same, but they've just scaled in size, and they're so conserved in their genetics and their body plan that they are actually still in the same one genus, even though they have a worldwide distribution. The smallest living varanid is brevicorda, Varanus brevicorda, the short-tailed monitor, and that's in Australia through the central desert areas. Our largest species in Australia is the parenti, again in central Australia. The largest living varanid is, of course, the Komodo dragon, found in parts of Indonesia. And the largest ever species to live was Megalania, that was over 8 metres long. A very impressive animal, would have been very great to see, but obviously very dangerous. Varanids are generally carnivorous. They have a very powerful chemosensory olfactory sense, which is made up of their tongue, their pointed tongue, forked tongue, sorry, which they plug into their Jacobson's organ in the top of their mouth. And there's also anecdotal evidence that they are intelligent. They're considered intelligent, even though there's been no really rigorous studies on that. But I can attest to the fact that they are very cognitively developed, having worked with them for a long time. Because varanids range in size and because of their life history traits and their diet, they hold really important roles in the ecosystems. They also can attain really high densities in environments, healthy environments like floodplains. This is because they engage with animals below them. They regulate some of the food web below them. They engage with animals kind of next to them on the food web and above them. Because they're engaged in so many ecological interactions, they're considered a keystone species, which means that when they decline, different parts of the ecosystem kind of collapse. So they're important to keep around, as I said, not just for themselves, but for the integrity of the ecosystem as a whole. I'd just like to give an example of this. Again, Panoptes that do attain really high densities on floodplains, and this was the subject of my PhD. And I measured the growth. So considering that these animals are very large, they actually grow really, really quickly. This is a growth curve. The bottom line is the female growth trajectory. The top line is male growth trajectory. The x-axis is age in days, and the y-axis is size in snout to vent length. Around the time they diverge is roughly when they're starting to reach sexual maturity, and that's around 200 days. And by 400 days, a male Panoptes is 500 millimeters SVL, so half a meter body length plus tail. That's a big animal, and that is fast growth. But how do they put on that much weight through the year? Well, they're generalist predators. Here are some of the species that they consume that we've got through their diet and scat and gut analysis. And they actually consume a lot of prey. Males will consume 2 to 3.5 kilos per month, and females 7, just under a kilo to 1.5 per month. And that is a lot of prey when you consider that these animals are active for 6 to 8 months of the year, and that in healthy environments you can have thousands of these animals present in the environment, as I have witnessed. So that is a lot of biomass to be taking out of the system and to be cycling nutrients through the system. You can imagine the hole that's left when they decline from toads. And this is an example of that growth. This is a young hatchling that I caught in November 2013, and by March 2015, so 2 wet seasons afterwards, he had grown into this impressive male. Goannas also have really interesting behaviours and personal lives. This is an example of a behaviour that we may measure. This is what you would consider a shy male. So me thinking I'm going to get a really good video of the release, no such luck. And this is a male of the same size. Obviously a lot more feisty. This is what we consider a bold male. Aggressive, scary male. I gave to my research assistants to release. Yeah, so goannas have really strong behaviourals that are actually consistent through their life, which was an interesting finding, something that really fascinates me. We measured lots of different behaviours of the animals that we were working with and saw that they were consistent through their life, and also that they correlated with each other, so that we could create groups of animals that had different personalities on the bold and shy continuum. And the interesting thing is that goanna's personality actually correlates with its behaviour, with its ecology in the field, and its mating, how many matings it gets, how big its home range is, and how many resources it can access. An interesting example of this is what happens in the wet season. Bolder animals, irrespective of whether they're male or female, will live closer to the rivers on the floodplain. The rivers are these really resource-rich areas, but they also have a lot of predation risk there. So the animals, bold animals, are living there. They actually get more resources, they get more matings, they grow faster, they have a bigger home range, but they're also exposed to predators. The shy individuals move away from the rivers in the wet season. They have less access to resources, they get less matings, but they also have less risk of predation. We know this because pythons, black-headed pythons and olive pythons, come and would eat our goannas. And we were able to then track the pythons that now had the transmitters of the goannas inside them. And what we saw was that the pythons were living up in the escarpments. They were coming down the rivers to hunt. They would catch goannas, sit there for a few days, digest, and then they would go back up to the escarpment and wait until they wanted to come back down again. So they were really honing in on those that resource of the river, which Panoptes does as well. So I'm not just a crazy goanna lady. They do have personalities, and I've proven it. Panoptes also has really interesting reproductive and nesting biology. They'll nest communally and they'll come back to the same nest year after year, even though for most of the year you can't even tell that it's that there is a nest or a burrow there. And they actually create these really deep helix-shaped burrows. I didn't discover that. That was Sean Doody and Simon Klulau, but it's a fascinating finding. Deep helix burrow with multiple clutches, egg clutches in the same nest, and down there they get to really stable temperatures. On the right you can see a data logger that I put onto a female. It's a thermal logger, and as she goes down into her nest you can see it basically flat lines for many days that she's at that stable temperature. But you don't actually need to go that deep to get those stable temperatures in the tropics. Only half a metre will do. So what this looks like is that these animals have evolved this kind of nesting structure, this complex nesting structure, for a predator that they probably doesn't exist anymore, that they don't need to go to this length to preserve their clutches. Dingoes and goannas and pythons maybe would be the predators for these, but they definitely don't need to go to those lengths. So it's really interesting that to be able to kind of see how old some of these life history traits are, and it makes sense for a group that has been around for so long. Another neat thing about the varanids in particular is that they have huge cultural importance for Indigenous peoples across Northern Australia and probably the rest of Australia. They are an important bush tucker, especially the large species such as Panoptes, but also Mertens. So there's a lot of creation stories around them, and creation stories really are the social compass for Indigenous communities about ethics and morals. There's also songlines associated with these animals which trace the ancestral pathways of these ancestors through the dream time, and those songlines still act as maps for navigation that people rely on today. And of course there's a lot of hunting knowledge and language associated with these species. So you can imagine how, the big gap that is left when these animals decline. For example, in some languages, there's upwards of 5 words for a goanna burrow. There's word for whether there's the burrow is old, whether it's new, whether there is a goanna currently in the burrow, whether the burrow is a refuge or a nesting burrow. So a really big impact when these species are gone. And that's an important thing for conservation to think about, is the impact that species decline has on Indigenous culture, and the fact that even if species exist elsewhere in the range, for those Indigenous cultural groups in those particular areas, if they've lost those animals on their country, then that's going to impact their culture dramatically. So we know that toads have an impact on a handful of species, and that those impacts ramify through the food webs, and it's important to do something about toads. What have we actually done? We've tried to eradicate them and control their populations through trapping and hand collection, releasing parasites, and also increasing interactions with native predators such as meat ants that can eat toads. That hasn't worked. We've tried invasion containment through fencing. Recently, there's a group that are looking into trying to create what they're calling a waterless barrier. There's a strip of land above the Pilbara and the coast, and they believe that if they can alter the water bodies there to stop water from being on the ground, freestanding water, which is what toads need to disperse and to breed, that they will technically be able to create a barrier a waterless barrier that toads could theoretically never cross in one wet season. So there's more work ongoing about that. Therefore, if that worked, it would slow the invasion, potentially stop it. And there's also impact mitigation. And that's really where my research falls in. So this is moving away from working with the invasive species towards the native animals that it impacts. And the main way we do that is called taste aversion. And I'm going to let Rick tell you more about that.
Our native animals eat large cane toads and die from the poison. When they eat small cane toads, they get sick, but don't die. So when they see more cane toads of any size, they don't eat them. They have learned to avoid cane toads. However, at the cane toad invasion front, there are only large colonising cane toads. They don't breed at the invasion fronts, and this is where they have the most devastating effect on native animals. Our research has shown that adding small cane toads at the invasion front can teach the native animals to avoid cane toads before the large fatal ones come through. Our native animals can coexist with cane toads, but only if they know not to eat them.
Yes, taste aversion. Teaching animals to avoid cane toads by giving them small doses of cane toad toxin. It's essentially a food poisoning response. All animals have the ability to develop food poisoning to avoid toxic substances in their environment, and we're just capitalising on that ability. My work was quite pivotal in the development of this strategy because we worked with wild animals and wild reptiles in particular. For my PhD, I worked at Ubulgari, which is a 16,000 hectare floodplain in the East Kimberley, very remote, only accessible by boat or helicopter. And it's a beautiful, rich floodplain, very biodiverse. It's a wildlife haven all through the year, but particularly in the dry season. And historically, it was a meeting place, a very important meeting place for all of the Indigenous groups across the Kimberley in the dry season to come and trade and do corroboree. And it sits on Balangara country. So I work closely with the Balangara community and the Balangara Rangers for the 3-year project that was my PhD. We lived on country for most of the year to catch the seasonal variation, and we just lived goanna life. We would go out and every morning catch goannas. We'd fit them with radio tracking devices, and then we just follow them. We'd take behavioural measurements, ecological measurements, and of course we were doing the taste aversion training. Essentially, we had a group of animals that we had fed small toads to that were small enough to make them sick but not kill them, and there was a group of animals that was control, that had not been trained, and we waited to see what would happen when the cane toads came through. Now, the reason that the metamorph toads are not fatal is because they rely on the toxin from their mother all through the early stages of the tadpole and growth and into metamorphosis. And the metamorph toads are not really toxic until they've developed their own glands themselves. And so that's the life stage that we worked with. But this is what it looks like in practice. This is Macca, a yellow-spotted monitor, and this is his first interaction with a cane toad. As you can see, he doesn't know that it's not something he should be eating. There's no cues, and he just gobbles it down. We would go out at night and track the animals to their burrows and place these arenas out the front and hope that they would encounter them in the morning. This is a subsequent interaction many weeks later. And you can see that he investigates the cane toad thoroughly. But then moves on. And we would repeat these trials with animals to make sure that the aversion was still present, or to test whether they kept their aversion over a long period of time. And this was all before the cane toads arrived. Once the cane toads arrived, that's when we saw the exciting results. What you need to compare here is the top line, which is the trained animals, versus the bottom line, which is the untrained animals. And what we saw was that once toad arrived, the trained animals lived in much higher numbers and for much longer over important periods of the year, aka the wet season or breeding season, than the control animals, which had all basically died within a few months of cane toads arriving. So training had increased the rate and length of survival for wild Varanus panoptes, which was very exciting. The research got a lot of attention in the media and elsewhere, and it was kind of paved the way for the rest of the research, because for the first time we'd shown that this could happen in the wild with wild animals. We know that most of the species that are heavily impacted by toads do have the ability to develop taste aversion in this way to cane toads, because there has been many studies done on them by many people, which is really exciting. And it's meant that taste aversion is now the most promising strategy that we have to mitigate the impact of cane toads. But what does that look like on the ground? Well, as Rick said, the invasion front is made up of really large toads that are fatal. Animals only have to mow the toad for 10 seconds before they die. And then you get the smaller toads, less toxic toads behind once the frontal toads begin to breed. So this is the natural situation, and what we need to do is swap it around. We need to bring the small toads to the front so that animals have an opportunity to learn from a non-lethal encounter with a small toad, and then remember that aversion when the big toads come through. You may have noticed that the bulk of species that are impacted by toads are reptilian. And so as herpetologists, you're probably asking, well, how does that benefit the next generation? Because a lot of reptiles, most reptiles, do not have parental care. They can't teach their young, so they can't pass it on. So how does that help the offspring? The strategy is designed to mitigate just the very first wave of the impact that comes through, which is the most devastating wave. It targets breeding adults because in subsequent years, when juveniles are born and are able to eat toads themselves, there will be natural opportunities in the environment for them because toads would have started breeding. So there will already be small toads in the environment. So it's just that initial impact zone that we're trying to buffer. We do have a realistic expectation of survival though. Not everyone will survive, as you saw in that graph, but when the situation is 0% survival, a 50% increase or even a 25% increase is better. And the thing is that for some species like panoptes, that fast, rapid growth, fast time to sexual maturity is really suitable for this strategy, because we buffer that initial impact and those animals survive through the wet season to lay eggs, which are then going to be babies in the following year where there's small toads around, but then they will also be having babies in the following year. So it really allows for a faster trajectory population recovery out of the impact zone. Now, I wanted to translate the taste aversion research into a landscape style strategy. There'd been so much put into it with all of the really important species. So after my PhD, Rick and I negotiated with a range of stakeholders to come on board for a big, ambitious taste aversion project that we would roll out ahead of the invasion front in the Kimberley. The diversity of stakeholders on our project is really impressive and exciting, because these organisations usually do not work, together or in big groups, because they've got their own things happening, projects happening. So to bring everyone together is great. We've got conservation organisations, the state government, the Kimberley Land Council, which is the peak Indigenous body, really important to have those guys on as stakeholders. We've got pastoralists, and we've got Matzo's Brewery, which is also a very important partner to have on a project that involves a lot of field work. So it's an impressive group of organisations, and we hope that it's going to be more of a model for conservation going forward, because it's very diverse, has lots of different stakeholders. We called ourselves the Cane Toad Coalition, and we've got a website there, which I'll also put up at the end. We work in the Kimberley region at the coalface invasion front. We work in areas of high biodiversity, where there are known populations of the predators that are likely to be impacted. So the Kimberley is the last biodiversity stronghold of Australia, and the toads are just marching through, and they should be finished that area in 3 to 5 years. So time is really running out. We use a number of cues and to elicit the aversion, and the cue we use depends upon the ecology and the hunting strategy of the predator that we're targeting. Varanids respond much better to metamorph toads, live prey that we breed and release, or that we collect and release. It also provides a stronger cue for them, that smell and that taste, that olfactory sensations coming in there. There are cane toad sausages that are made as well, which are cane toad flesh with a nausea-inducing strong salt in them. And they are used to target quolls, because quolls live in rocky country, difficult to deploy metamorphs into that country. I don't do the quoll work myself, that's colleagues at the department and Melbourne Uni. I know it sounds crazy to be releasing an invasive species to mitigate the impacts of the same invasive species on native predators, but I like to think about it as counterintuitive conservation. It's an innovative strategy. It's an innovative strategy that is the culmination of decades of research on all the other options, basically. It's also a move away from traditional invasive species management, and it's actually really exciting. But we do have checks and balances. We make sure that deployments happen in the same season as the toads will arrive. And this is so that metamorphs don't move away from water bodies, increase the invasion, which is really unlikely if you've actually lived through the invasion to see how much the environment's just saturated with adult toads. But it's more about kind of public perception and making sure that everyone feels comfortable with it. But it's also about ensuring that the cue is as strong and fresh in the minds of the predators as possible, so that it translates across to the adult toads when they arrive. We time the deployment seasonally, again, to limit dispersal, because we also don't want the metamorphs dispersing away from the water bodies. We want them there in high densities so that the predators are more likely to encounter them. We monitor all of the populations before we go into an area, do the deployments, and then monitor afterwards. We also look for off-target species and any negative impacts, but we haven't found any as yet. These are just a few of the species that we work with. Water monitors are a group that are impacted in some areas and not others, or they seem to be recovering in some areas. A lot of kind of artificial areas, artificial drainage lines and things the population seem to recover, whereas in a lot of the wilder places, they don't. And I think it's because they live in rocky habitats that, if it's a small water system, for example, then, they're flooded with toads, and there's less resources in the environment. We work with them in the North Kimberley, around Mitchell Plateau and those areas. We work with Mertens and Mitchells. And we're actually trialling both metamorph toads, or we call them teacher toads. We're trialling teacher toads as well as sausages, because it would be very beneficial if we could just fly along with helicopters and drop sausage baits would be a lot easier. It's quite time consuming to do the breeding and deployment of metamorph toads. There's a lot of things that can go wrong with the process. Just it's a big process. This is a picture of a happy Mertens in Transact at the Mitchell Plateau. Populations up there are really healthy and really widespread demography, lots of hatchlings every year. It's a beautiful country to work in. We do this project with Wonderful Gambera, who are, Rocky Gorge, freshwater people in this area anyway. And yeah, they know Mertens, they know Mitchells, they know water monitors. They're very important in their culture. They know the environment. So they're a pivotal part to us rolling out the strategy there. We are targeting freshwater crocodiles in some places. The Kimberley has been identified as an area with multiple vulnerable populations of freshies, and that's because water bodies and water systems are quite isolated and they dry out. Some of them dry out a lot in the dry season. So you get a situation where there are huge numbers of freshies in these fairly small water bodies. There's competition, there's strong resource depletion in those water bodies for extended periods of the year, and those water bodies are also likely to be a source for cane toads through the dry season as well when they get there. And these populations are potentially genetically distinct from each other, so they're important in that respect as well. Places like Windjana Gorge, that's going to be one of our main study sites this year. The way that we're targeting crocs is with apparatus that we hang over the water with baits on them. And the research at the moment is looking at where they take baits from. Do the baits need to be over the water? Can they be on the edge? Can they be up on the bank? Because we do see crocs coming up onto the bank. But also, how can we limit offtake for species, nuisance species that we don't want to take the baits before the crocs do? An interesting finding is that we're seeing crocs in Kununurra, where we've been testing the apparatus, are showing strong aversions to the toad baits versus the control baits. And that reflects a general behavioural shift that we see behind the cane toad invasion with many species to an avoidance of cane toads, which is obviously allowing persistence. Here's a picture, a snap of a video of a croc in the bottom right-hand corner there, reaching up to take a bait. That's a chicken neck. They do eat baits at any time of the day, as well as but they do seem to take them from the water most. The bank edge they will take from as well, but when the baits are on the banks, there's just too much offtake from non-targets, gyners and raptors and things. And that's just a photo of a little sneaky croc coming up and stealing a Mertens bait from Mitchell Plateau there. It's always funny seeing them walk around like little dogs on the land like that. And of course, a key species that we're targeting is Varanus panoptes, the floodplain monitor. Most of that work happens in the southern Kimberley, around the Fitzroy River drainage area. There's 5 different Indigenous communities that come together at Fitzroy whose land joins there, so an important species. An interesting finding from work with panoptes is that just like they have individual personalities that dictate different parts of their ecology, individuals also have dietary preferences that aren't necessarily related to their sex or their size. We found, for example, that animals that eat frog, and also there's correlation. So animals that eat, this graph is showing frogs in scats. Animals that eat frogs also are more likely to eat lizards. That's a correlation, and there's other correlations. But what we've seen is that animals that eat frogs are also more likely to die from cane toads in that initial wave, as you would expect. But then an interesting flow on from that is because those animals that eat frogs, die from cane toads, also have preferences for lizards, especially this Amphibolurus species, that has caused a spike in the populations of these lizards that we see after cane toads move through an area. And so that's just more evidence of the kind of regulatory role that Panoptes has in the environment. I'm just going to take a sidestep now that I've finished all of the conservation research component to talk about collaboration in science. I think it's really pertinent in communities like herpetology communities, where we've got a range of people of different skill sets and knowledge bases, huge, like rich knowledge, and the importance of recognising that. I work with Indigenous rangers through my PhD and all through the current project. They're a pivotal part of that project, of the success of that project. And I just wanted to tell you a little story about how that relationship can play out and how science can be influenced if you're flexible and open to new knowledge sets. So as I said, we would catch goannas in the morning, and the teams were set up to have an Indigenous ranger and a biologist. And we had multiple teams that would go their separate ways and hunt for goannas in the morning. So the ranger and the biologist in the team would have the same opportunity to see the same goannas each morning. But what we found was that rangers were able to spot goannas from further away, in denser vegetation, in shade, and that were stationary, versus animals that were moving in the open, but in the sun, and from close up, which was more what the scientists would see. And the scientists are professional herpetologists, including me. So that was kind of a surprise, but not really if you've worked with rangers. They have amazing skill sets, really impressive. So that was a pretty cool thing to actually be able to quantify. As I said, we quantified the behaviour of individual lizards as well through the life of the study. And what we found was that animals that rangers spotted were also shyer. This is a boldness score here. They were also shyer, which kind of makes sense if you think about how they were spotted. In the cryptic environment situation by rangers, they turned out to be shyer animals. But the real kicker to this part of the project was that even though rangers and scientists caught the same number of animals that ended up becoming trained with the taste aversion methodology, once the cane toads arrived, the animals that the rangers spotted actually learnt the methodology better and survived in high numbers, whereas the animals that the scientists spotted may have learnt the methodology before the toads got there, but then either when the toads got there or sometime afterwards decided to eat a toad, probably because they're the bolder, kind of less risk-averse animals. But what that meant was that the animals that the rangers had spotted actually drove the significant result of the taste aversion study. So if we hadn't worked with the rangers, we wouldn't have collected this group of animals, we wouldn't have the data that we have, we wouldn't be sitting here delivering the largest cane toad mitigation strategy that has been delivered to date, and having all of the positive ramifications that has. That's just a story that I wanted to share because I really respect my Indigenous collaborators and couldn't do this work without them. But B, it's also important to strive for diversity in science and in research. And I think this is really good proof of what can come of that. And lastly, an outcome of all of this work is that we will be able to apply what we've learned to other systems, which is really exciting. We've worked for decades on trying to mitigate cane toad impacts and tried so many different things. And where we're at now is a position where we can have a really meaningful conservation output and apply that to other places like Madagascar. They are experiencing their own invasion by another buffonid species, the Asian black-spined toad, and it's in its early stages, but they do also have a suite of vulnerable apex predators that have no resistance to the toad toxin and are likely to experience severe declines from that invasion. So we're really hoping to work closely with people over there to be applying what we're learning directly to that study system, instead of them having to wait for the same amount of time that we have. Thank you very much. It's been great to talk to you today about my research, and please get in touch with me if you have any questions or ideas, and also please check out the Cane Toad Coalition website. It's got all the information about what we do, who we work with, and yeah, I'd love to hear from you. I'm always up for talking herbs. Thank you.